Hi guys, we're back again, and today we are going to talk about movement of particles in and out of the cell. The movement of molecules in and out of the cell, there are two basic types. One is the active and one is the passive. Well, active of course needs ATP, all right, and does not need a concentration gradient. I'll explain this in just a minute, okay? Passive does not need an ATP but needs a concentration gradient. What's a concentration gradient? Well, here's a cell. There's lots of food outside, little food inside, there's more out here, there's less inside. Well, this is a difference in concentration gradient. Passive transport will mean more to less, but an active transport, it is going uphill. Imagine you're going uphill. Of course you'll have to pedal harder. You're on a bicycle, you're going to pedal harder. You need more ATP. Now what if you are wanting to pump this less to more toxic waste products inside the cell, you want to throw it out? Well, if you want to throw it out, of course you'll need ATP. So please note the difference between active and passive transport. Alright, the first type of passive transport that we are going to be doing is of course simple diffusion. So in simple diffusion, we definitely need some form of channel protein, and in simple diffusion the particles are going to move from more to less. You really need just channel proteins. The particles are going, there's more here, there's less here, and of course no ATP is required. There's more here, there's less here. So particles are going to move through this channel protein from the outside to the inside. Once again out here, as you can see that there's more here, less here. As long as there's more and less, flow will take place. As soon as the particles become equal in amount, there'll be no net flow. Why? Because now the amount is become the same. Okay. Facilitated diffusion is more complex. It is more sophisticated. For a facilitated diffusion, which is the second type of diffusion, again, we do not require ATP. But here what happens is we require carrier proteins. So carrier proteins are going to be very specific, bind to certain molecules. As soon as they bind, they are not only going to transport it, but they're also going to change their shape. So as they change the shape, they are going to transport specific molecules from outside to inside or inside to outside. This would be an example of facilitated diffusion using carrier proteins. We do not need an ATP. So once again, passive type of transport, the first one we talked about is diffusion, and diffusion is of two kinds, simple, and the second one is facilitated diffusion. Okay? The third type of passive transport is osmosis. Osmosis is the movement of water. Note the difference between diffusion and osmosis. Diffusion is the movement of any particles. Osmosis is the movement of water alone. So it's safe to say osmosis is a type of diffusion, but all diffusion is not osmosis. All right? So movement of water through a selectively permeable membrane from a region of higher concentration to a region of lower concentration. Now we are using a new word, solute. But even before we go on, let's take for example salt, and I'm going to mix it in water. So I'm taking salt, I'm mixing it in water, and I'm getting salt solution. Very simple, right? Salt would be my solute. Water would be my solvent. So what I'm saying is, osmosis is the movement of water from a region of from a region of lower solute concentration to a higher concentration of solute. So here, let's say for example here, you have lots and lots of solute. So this is very salty. Here there's little solute, this is less salty. So this is region A, this is region B. I've separated the two regions by a semi-permeable membrane. Notice water is going to move from here to here, low solute to high solute. Another example, take a bean, take a pea, take for example a raisin, put it in water. This is plain simple water. I'm soaking my bean. Well, what will happen the next day? The next day the bean would have increased in size. Why would have it increased in size? Water has gone in. What's the process by which water is going in? Well, of course, it is by osmosis. Well, why does water enter through osmosis? Once again, here, this is water, no solute. This is a bean. This is a raisin. Surely you know raisin is sweet. It's a living cell. Cells have 1% solute. So therefore, low solute, high solute, less sweet, more sweet, less salty, more salty, water is going to go in, and as a result of which your bean will be nice and swollen the next day. When we come to osmosis, we talk about at least four terms. Osmotic pressure. Osmotic pressure is the force. This is the force with which the water, which is going to drive the water from a region of low solute to a region of high solute concentration, right? We talked about this. And more the difference between the solute concentration, that means if this is very salty and this is very low salt concentration, more the osmotic pressure. So more difference in solute, more the osmotic pressure. In this term, we'll talk about hypertonic, isotonic, hypotonic in my next slide. Here are six scenarios, excuse me. So first we are taking an animal cell, a red blood cell, and we are putting it in plain simple water. So here's my red blood cell, I'm putting it in water. Well, this situation, remember, RBC is a living cell. It has 1% solute salt concentration. So this is plain simple water. Well, what do you think will happen? Water will enter, just like water entered a bean or a raisin, etc. Water will enter, and my RBC is going to increase in size. If it increases in size, it is going to burst. This situation would be a hypotonic situation because inside the cell is 1%, outside the cell is lower than 1%. This would be hypo. Hypo means less. Iso means same. I'm taking my RBC and I'm putting it in 1% salt concentration. So inside is 1%, outside is 1%. Water is going to go out, water is going to enter, same amount of water is entering and leaving. So the cell size will remain the same. This is an iso. Iso means same. Tonicity means concentration. So isotonic concentration, the cell size will remain the same. In a hypotonic concentration, water will enter and eventually the cell will become bigger and bigger and bigger and finally it will burst. In a hypertonic concentration, I'm taking my RBC and surrounding it in a very, very salty condition, like for example 80% salt concentration. In that case, remember this is a living cell, it has 1% salt concentration. So water is going to leave the cell. Water is going to go from low solute to a high solute concentration, less salty to more salty. So the cell will become shriveled in size. It has a cell membrane, so cell size is going to be very dramatic. In a plant cell, it is not going to be that dramatic because the plant cell has a what? It has a cell wall. Okay, so in an isotonic condition, when you're putting 1% outside, 1% inside, water's entering, same amount of water's leaving, the cell size or the turgor pressure will remain the same. Just hang on, I'll explain turgor pressure. Here, when I'm putting the cell in a hypotonic concentration, that means I'm putting an animal cell in just simple water, in a very, very weak solute concentration or plain simple water, water is going to enter the cell. Look at the central vacuole. It's going to become big, full of water. It is going to push against the cytoplasm. So cytoplasm is pushing against the cell wall. Cell wall is pushing inside. In this condition, the cell is tight, the cell is taut, and we say, oh, the cell is turgid. The cell has very high turgor pressure. This is very important for plants to remain erect. Okay, this is why this is needed for the plant to remain erect. Now, if the cell is surrounded by a hypertonic concentration, that means there is very high salty water outside the cell, well, water is going to leave the cell. Central vacuole collapses, cytoplasm collapses. No longer is the cell tight or taut. No longer is the cytoplasm pushing against the cell wall, so the cell will become flaccid. And this is what happens when you do not water a plant. The plant has collapsed. It is saying, oh, I need some water. It is no longer going to be erect. It's going to be flaccid. So since we're animals, of course I'm interested in these conditions, right? So here you want your cells to be in isotonic condition. You do not want your cells to be in hypotonic condition. You do not want your cells to be a hypertonic concentration. A hypotonic concentration would be, for example, when you flood the cells with water, like you binge on water. So this would be a hypotonic concentration. You don't want that. Moving on. We are talking about active transport. Active transport requires ATP and carrier proteins. Well, ATP we understand, okay? We are going uphill. If you are going uphill, if you are going from low concentration to a high concentration, you absolutely need to pump ATP. You are going to use the ATP, pump these molecules from low to high. But you also need carrier proteins. Carrier proteins are going to be very specific, bind to specific molecules, change its own shape as it moves the molecules into the cell or out of the cell. It really doesn't matter. So once again, to repeat, active transport needs ATP and carrier proteins, does not need a concentration gradient. It can go against the concentration gradient. We understand that, don't we? This is conc. All right, what if you need to do bulk transport? Bulk transport is endocytosis. Endo means going into the cell. Cytosis means cell. So here what happens is cell will engulf particles and form vesicles. So here this could be receptor mediated. I'm going to cut out the word pinocytosis and write endocytosis. So here receptor mediated endocytosis is very selective and efficient. It's not just going to take in any molecule. It's going to be very specific. There are two types of endocytosis. Pinocytosis, when it engulfs a liquid, as you can see here, it is engulfing liquid, and phagocytosis, when it engulfs solid. Here, as you can see, it's engulfing a solid body, and this is typically seen by WBCs as they engulf any pathogen, any foreign body, as they swallow a pathogen. Pathogen, you don't need to know what it means, but basically it means any organism that causes a sad story. Pathogen would be a disease-causing organism. So here, this is phagocytosis and that is pinocytosis. So this is exocytosis. If exocytosis is the reverse of endocytosis, you are trying to get something out of the cell. In that case, substances, these vesicles are going to fuse with the cell membrane and it's going to release outside. This is how secretion takes place. This is how neurotransmitters are released. This is just a table format, differences between passive and active, what it needs and what it does not need, what it includes and what also this includes. So I hope that you have understood. If there are any questions or concerns, please call me, please talk to me, please ask me.